Assalamu alaikum. Today we are going to talk about fertilization. Phases of fertilization. What is fertilization? Fertilization is a process when male gamete fuses with the female gamete and a diploid zygote is formed. Male gamete and female gamete both are haploid, containing single set of chromosomes, but when they fuse, the zygote is formed and zygote is diploid, having double or two sets of chromosomes present in the form of pairs. Usual site of fertilization is the uterine tube. If the fertilization does not occur in the uterine tube, the ampulla of the uterine tube, the sperm is carried towards the uterus, and finally the sperm is degraded in the uterus. Fertilization can also occur at other places, but it does not occur at the body of the uterus. Chemical signals are secreted by the secondary oocyte and the follicular cells. These chemical signals guide the sperm, the capacitated sperms, towards the uterine tube. Fertilization is a complex sequence of events that starts from the sperms attacking the secondary oocyte containing a layer of Zuna pellucida and the corona radiata. These are the follicular cells that form the corona radiata, outer covering of the ovum. So the process of fertilization starts from the sperm attacking the oocyte and ends at the first metaphase of the mitosis. Now we will see the individual phases of fertilization in detail. First of all, dispersal of the corona radiata cells means the follicular cells surrounding the secondary oocyte occur. The dispersal is considered that the acrosome of the sperm contain an enzyme called hyaluronidase that degrades the follicular cells and provides the passage to the sperm to enter into the secondary oocyte. But there is thought that there may be tubal mucosal enzymes and the movement of tail of sperm is also important in the penetration or in the dispersal of the follicular cells and providing the passage to sperm. Here you can see that it is starting to come into the corona radiata. Finally it tries to put itself inside and make the passage, secreting the hyaluronidase enzyme, tubal mucosal enzyme, and then also movement is also moving or providing the motility to the sperm, and then the sperm is finally go into the follicular cells and corona radiata. Next stage is the penetration of the sperm to the Zuna pellucida. This is Zuna pellucida. So the penetration occurs by certain enzymes, esterase, acrosin, and neuraminidase. You will study in biochemistry what are neuramine and neuraminidase enzyme. Esterase, acrosin, and neuraminidase. This causes the lysis of the Zuna pellucida. Sperm is penetrating the Zuna pellucida by lysis. The main important enzyme is acrosin enzyme. Acrosin is the main enzyme that take part in the process of lysis of Zuna pellucida. After penetration through the Zuna pellucida, the next phase is called the Zuna reaction. What is Zuna reaction? It is a third stage. Second, it was the first stage. Zuna reaction, when the one sperm is able to cross the Zuna pellucida and enter the secondary oocyte, it causes specific changes in the Zuna pellucida. That Zuna pellucida becomes impermeable to other sperms to come inside the secondary oocyte. You can see here, this is the one sperm that is very lucky and has entered the secondary oocyte, and the other sperms are unable to enter. However, if they were able or not, the main reason is the Zuna reaction. Now Zuna pellucida is not allowing other sperms to come inside. The changes can also be due to the enzyme secreted by the perivitelline space. You can see here, this is the Zuna pellucida. Here is also a little space of the membrane which is called the perivitelline space. The perivitelline space secretes special enzymes that cause the entry of the other sperms impossible. So this is the Zuna reaction. Remember one thing, that Zuna reaction, when the sperm entered the Zuna pellucida, is made impermeable for other sperms. One change. The other change is perivitelline space. The secretions or the enzymes etc. released from the perivitelline space causes changes in the plasma membrane of the secondary oocyte. This is the actually perivitelline space is the plasma membrane of the secondary oocyte. So the secondary plasma membrane of the secondary oocyte is changed in such a way that the other sperm is not allowed by the plasma membrane to enter the secondary oocyte. The next step is, the fourth step is fusion. Fusion of what? Fusion of membranes. First of all, membrane. At this point, when the sperm was trying to enter the secondary oocyte, the plasma membrane of the secondary oocyte is fused and the sperm enter into the secondary oocyte. But still at this point, the sperm has its mitochondria and cell membrane intact. You know that at this point the nucleus is decondensed. Nucleus is present in two forms. It may be condensed or decondensed. Decondensed and decondensed. Decondensed form is when the cell is dividing. When the cell is in the non-dividing stage, it is condensed. So at this point, still the secondary oocyte is at metaphase 2, meiosis 2, metaphase of meiosis 2. It is in the decondensed form. Whenever the sperm entered, the second meiotic division started its completion. The female nucleus from decondensed to condensed form, and it formed the pronucleus, female pronucleus. So it formed the female pronucleus. After the female pronucleus is formed, the male nucleus, means sperm nucleus, also enlarged. The acrosome has been lost, and the cell nucleus enlarges and form the male pronucleus. The cell which contains two pronucleus, one is the female pronucleus, other is the male pronucleus. This cell is called otid. Otid. Now in the otid, the two pronucleus come towards each other and they start fusing with each other, and also fusion occur, and also the breakdown of pronuclear membrane. They will form a zygote nucleus. Whenever zygote nucleus is formed, it hurriedly moves and forms a decondensed stage, and in the decondensed stage the mitotic spindle, cleavage spindle formation occur, because at this stage the fertilization has ended and the zygote is formed. This is the zygote. Zygote is unique in its characteristic that it contains a genetic makeup that is the combination of two cells, two individuals. One was the mother and other was the father. So this type of inheritance is called biparental inheritance. Biparental inheritance. So it was all about the phases of fertilization. We can summarize that phases of fertilization start from when the sperm was trying to cross the hurdle. The hurdle was the corona radiata. The corona radiata, and the acrosome releases hyaluronidase enzyme that degrades the hyalurin and tubal mucosal enzymes, and also the movement of tail of sperm. These factors are important in the dispersal of the dispersal of the corona radiata. The next stage was the Zuna pellucida penetration. After the Zuna pellucida penetration, the third stage was the Zuna reaction. The Zuna, when the sperm entered, the specific changes occur in the Zuna pellucida, that Zuna pellucida become impermeable for the entry of other sperms. This was the Zuna reaction. In the Zuna reaction, one more event occurred, that the perivitelline space, the plasma membrane of the secondary oocyte releases special type of structures that make the plasma membrane impermeable for the passage of the other sperms when one sperm has entered. Next was the female pronucleus formed, fusion of membranes. Secondary oocyte, when the sperm is trying to enter, secondary oocyte has fusion of their membrane and allow the passage of sperm inside the cytoplasm, but the sperm contains its acrosin, its membrane and its tail. Then the tail start disintegrating and the smaller nucleus of the sperm got enlarged, and it formed the male pronucleus, and female pronucleus is formed by the decondensed to condensed nucleus of the female. Now the two pronucleus present in a structure, the cell is called the otid. Then the fusion of these structures, these two pronucleus occurs, and the two pronucleus, then the membrane of the two pronuclear membranes start degenerating. This is the completion of the fertilization. But just after the completion of the fertilization, the the zygote start cleavage, and the cleavage spindles are formed, and further the cleavage occurs. We will discuss the cleavage in our next lecture.